Hi everyone. We are now in our second lecture for chapter 7. We're going to be looking at some smaller structures today than we looked at last time. This lecture is going to look at the microanatomy of bone. We're going to be looking at the histology of bone or osseous tissue. Again, this is going to be another shorter lecture, kind of like we had last time. And after today, we will have one more lecture on this chapter. So let's start with our not really attendance questions. First, what is found in the medullary cavity of a child? And what about an adult? And then, what type of bone is found in the walls of the diaphysis? So go ahead and try to answer those. We're not going quite as far back as we usually do. These actually both came from the previous lecture. And hopefully you've paused the video now and tried to answer these on your own. But let's see what the answers are. What is found in the medullary cavity of a child? This is also called the marrow cavity. So here in a child, we find red bone marrow. But as the child reaches adolescence, that red bone marrow is no longer needed, so we only find red bone marrow in a few places throughout the skeleton. And those medullary cavities are, instead of being filled with red bone marrow in an adult, they're filled with yellow bone marrow. And remember, red bone marrow actively makes blood. Yellow bone marrow is fatty. It's for energy storage. And we'll re-mention that again today. Next, what type of bone is found in the walls of the diaphysis? The diaphysis, remember, is the shaft of a long bone. So we're looking at what makes the walls of that shaft. This is where we find compact bone. We find compact bone a few places, but this is generally what we use as our example of where we would see compact bone. So as I said, we're looking at some smaller structures today. We're down at the cellular level, and even in some cases, we're going to get smaller than the cellular level. So let's start talking about some of the types of cells that we will see when we look at a bone under a microscope. And different types of bone cells tend to have osteo in the name, because remember, osteo is bone. So the first cell that we're going to look at is called an osteogenic cell or an osteogenic stem cell. And it is a stem cell. Remember when we looked at skin, we had the stratum basale, that was a layer of stem cells that produced the cells in the skin? Well, osteogenic cells are stem cells that produce most of the cells that we find in bone. So osteogenic cells, remember we mentioned them when we talked about the periosteum and the endosteum. Those were both places where we would find osteogenic cells. And osteogenic cells, they are stem cells, and when they divide, one cell remains an osteogenic cell, and the other cell becomes something called an osteoblast. We've seen blast a few times now. Remember, blast means to make or to give rise to or to birth. So an osteoblast is a cell that makes bone. And that process is called osteogenesis. Osteogenesis. And we're going to see a little bit later that while we can say they are making bone, they are not making bone from scratch. We don't really produce bone, we convert cartilage into bone. So we're going to be looking at that a little bit later also. But osteogenic cells produce osteoblasts. Osteoblasts perform osteogenesis. They make bone. Now, as they are making bone, what they're doing is they are calcifying some softer tissue and making it hard like we imagine bone is. And while this is happening, some of those osteoblasts actually get trapped. They surround themselves with that hardened bone. And once they are trapped and completely surrounded by that bony matrix, they go through some changes. They become something called an osteocyte. And an osteocyte is a mature bone cell. Osteocyte is a mature bone cell, and it used to be an osteoblast before it was trapped. So what do osteocytes do? They basically just maintain the small area of bone that they are surrounded by. They maintain existing bone. And they're actually going to be trapped, like I said, surrounded by bone, but they're sitting in a small pocket, an open space. And that pocket that an osteocyte sits in is called a lacuna. More than one is called a lacuna, or called lacunae. One is a lacuna. So a lacuna is the pocket that an osteocyte sits in. Now we also see lacuna in cartilage. So really this is just a pocket that holds a cell. Now in bone, the osteocyte sits in the lacuna, but it actually has some small extensions of its cytoplasm that reach out. And the nearby osteocytes also reaching out, those extensions of the cytoplasm will actually connect with each other. And gap junctions will be formed between one osteocyte and the next. So that allows these osteocytes, which are basically trapped here, to still be able to communicate and transfer materials with its neighbors. Now these extensions, like we could see right here blown up, but these extensions that reach out and connect to neighboring cells are called canaliculi. Canaliculi. That really just means small canal. So these canaliculi are extensions of the cytoplasm that connect neighboring osteocytes. And where these canaliculi connect with the neighboring osteocytes, we find gap junctions so that the cells can exchange material and communicate. Now there is another type of bone cell, but it comes from a different source. The same stem cells that give rise to blood cells also give rise to another type of bone cell called an osteoclast. When we see clast at the end of a word, it means to break down, to crush, to destroy. So osteoclasts are responsible for something called osteolysis. LYS, remember, means to split or to break apart. So here we are breaking apart bone. And this isn't always a bad thing. We are constantly breaking down bone and building new bone. So this isn't a bad thing. But the cells that break down bone are called osteoclasts. And one side of the osteoclast has a lot of microvilli. And this side that has all those microvilli is called the ruffled border. And this is not unique to an osteoclast. There are lots of places in the body where one side of a cell has a lot of microvilli, and when that's the case, it's usually called the ruffled border because under a microscope, it looks a little bit ruffly. But this ruffled border secretes enzymes like lysozyme and a few other enzymes that are responsible for breaking bone down. So now getting even smaller, getting smaller than bone, we're going to start looking at the components of the matrix. So the matrix, the area outside of and between the bone cells. And when we look at the matrix, there's two main classes of components. Some of those components are organic. So they, either were carbon-based or they are from a living source. And then there is inorganic, so not carbon-based, and these are going to be things like minerals, so not from a living source. The organic component, the organic matter, let's look at something called the osteoid. And the osteoid really is all of the organic matter collectively. Osteoid, by definition, is the non-mineral part of the bone matrix. And in the osteoid, we find things like proteins, like collagen. Remember collagen we saw in the skin, and in lab you saw it in a lot of the connective tissues? Well, collagen is also found in bone. We find a lot of different types of carbohydrates, like proteoglycans. So these are things like protein and carbs. They make up the osteoid, they make up the organic part of the bony matrix. Now, on the inorganic side, here we have a whole bunch of different minerals, things like calcium, magnesium, phosphate, and collectively we will call these mineral salts, we'll call them hydroxyapatites. Hydroxyapatites. These are calcium phosphate salts. But there are other minerals, like I mentioned, things like magnesium. And as we said before, remember, bone is like a storehouse. It's like a bank for minerals. So this is just kind of a way of saying minerals as opposed to organic matter. You do not need to know all of the individual minerals. Okay, so let's change gears a little bit and start looking at the structure of bone. And here we have the structure of compact bone. And when we look at bone under a microscope, which is what we see down here in the bottom right, it kind of looks like tree rings. And those ring structures are very, very easy to see through a microscope. And here we have kind of the cartoonized version to show you what it is that you're seeing. And what we've done here is we've taken the shaft of a long bone and we've cut a wedge out of it. And I wish that there was a little bit of a different view of this, because a lot of students, I think, get confused with what they're seeing. These rings right here are microscopic. I think a lot of students look at this and they think that this hole right here is the medullary cavity, and it is not. The medullary cavity is actually what would be in this area right here. It would be very big in this view. Right here, this wedge that we've cut out is a very small portion of bone. It's actually up here. This is what makes the wall of the medullary cavity, or the edge of the diaphysis. So we're looking at this microscopically. And each of these large ring-like structures, that's what we see down here in this image, and that's what you looked at in the microscope in lab. So here we can see one of those large ring-like structures. Here's another one next to it. Here's another one. Here's one that's been pulled out so we can see the layers. Well, each of these large ring-like structures is called an osteon, or sometimes you will hear it called a Haversian system. An osteon or a Haversian system. Now, an osteon, Haversian system, is the structural unit of compact bone. The structural unit of compact bone. And one osteon actually runs the length of the diaphysis. So we can see these rings here, they're pulled forward so that we can kind of see their structure, but they run the entire length of the diaphysis. And same with the neighboring osteon, it runs the entire length of the diaphysis. And these layers that run around making the walls of that osteon, those are called concentric lamellae. Lamelle means page or sheet. So these are like pages of a book, or they're really like the rings of a tree, I've always thought. But these are the concentric lamellae. One layer, the layer just outside of it, the layer just outside of that. And here we've pulled some of them out. We can see the innermost concentric lamellae, then the one just outside of that, and the one just outside of that. So concentric lamellae surround one osteon. The neighboring osteon will have its own set of concentric lamellae. And right in the middle, we have this hole. In the microscope, on the slide, it looked very dark. You couldn't really make out much of what was inside. But that is called the central canal, or the Haversian canal. And the Haversian canal, or the central canal, is where we find the blood and lymph vessels, and in some cases, nerves. The blood and lymph vessels, and in some cases, nerves. And those blood vessels and lymph vessels and nerves supply all of these cells of this one osteon. So this osteon, we can see the vessels for it. Here's the neighboring osteon, we can see the vessels for it. So now think back a moment ago when we were looking at the bone cells and how the osteocytes became trapped and they held on to each other. They were in contact with each other with those canaliculi. And we could see that down here, the dark spots. These are the osteocytes, and these lines, those are the canaliculi. Well, the blood vessels here can supply with oxygen and nutrients and pick up waste only the cells in the innermost lamella. The cells that are further out, well, this tissue is very hardened. It would not be easy for nutrients and oxygen and waste to diffuse across. So instead, the cells that are closest to the Haversian canal, or the central canal, exchange with the blood vessels. But as nutrients are dropped off, those nutrients can pass through one osteon to the next osteon, from one osteocyte to the next osteocyte to the next osteocyte through those canaliculi. The canaliculi allow passage of things like nutrients and waste. Now, we also have something called Volkmann's canals, or perforating canals. And those Volkmann's canals, or perforating canals, actually run across the bone, and they connect neighboring Haversian canals. So we said an osteon runs the length of the diaphysis, and so do the blood vessels within the osteon. Now, the neighboring osteon, we can see here, is connected crossways. And this connection is called a Volkmann's canal, or a perforating canal. And when we get close to the edge of the bone, the Volkmann's canal actually exits the bone through one of those small nutrient foramen that we saw earlier. So the blood vessels and the lymph vessels enter the bone. They travel through a perforating canal, or a Volkmann's canal. They get to a central canal, pass through another Volkmann's canal to a different central canal. So all of these structures are connected to each other. Now, if we look at the edge of the bone, we can see that there are some layers here also. But these layers don't look like they are surrounding an osteon like these concentric lamellae did. These layers actually run all the way around the diaphysis of the bone, and these are called circumferential lamellae. Circumferential lamellae run all the way around the entire circumference of the diaphysis. And the final type of lamellae that I want to draw your attention to is, well, if we have a bunch of circles and we put the circles next to each other, well, circles don't fit neatly next to each other. There's going to be some gaps in between. And those gaps are filled with special types of lamellae called interstitial lamellae. Interstitial lamellae do not belong to a single osteon. They fill in the gaps between osteons. Now, briefly, let's talk about spongy bone because it's got a completely different makeup from compact bone. Here we can see the spongy bone has kind of this red tint to it in this picture, and it looks completely different from the edges here where we would find compact bone. So spongy bone is arranged in these kind of spiky looking structures here. And if we looked at them really closely under a microscope, some of them would be kind of flat, and those are called trabeculae. Some of them would look like little tiny rods, and those are called spicules. But spicules and trabeculae are the structural units of spongy bone. And microscopically, they do have something that kind of resembles an osteon. There's some heavy modifications to it that we will not get into, but it's kind of like an osteon. And as we mentioned a couple of times now, bone marrow. And there are two different types of bone marrow. At birth and through toddler and early childhood, all bone marrow is red bone marrow. Red bone marrow is sometimes called myeloid tissue. Sometimes it's called hematopoietic tissue. But this is where we make blood. That process again is called hematopoiesis. Red bone marrow performs hematopoiesis. It makes blood. Now, as we age, as we move from childhood, adolescence into adulthood, some of that red bone marrow, the red bone marrow that's in the medullary cavities, goes through a change and it is replaced with yellow bone marrow, which is very fatty. And instead of hematopoiesis, it's going to be a site of energy storage. And we will wrap up with talking about how bone grows and what causes bone to grow. So first we're going to look at how bones grow in length. We're going to look at bone elongation. And what we're looking at here in this picture is the epiphyseal plate. This is the growth plate. And this is the growth plate of someone who is still actively growing. So they are still actively getting taller. In an adult, it would not look like this at all. And what we're looking at is this area right here, right where the epiphysis and the diaphysis meet. This is the epiphyseal plate. And under a microscope, the epiphyseal plate has very distinct regions. And over here, they're kind of color-coded. But you can, even without the color coding, you can see that different regions look very different. So let's see what it is that we're looking at. At this end of the bone, we are going to start with the side that's closer to the epiphysis and work towards the diaphysis. At the other end of this bone, we would see the exact same thing, but it would be upside down from what we see here. So in the top of this picture, this is closest to the epiphysis, the end of the bone. And as we're going to move down this picture, we are moving more towards the diaphysis, the shaft of the bone. So up here at the top of this image, we find hyaline cartilage, and this is called the zone of reserve cartilage. The zone of reserve cartilage is just hyaline cartilage. And now moving towards the diaphysis, the next area that we come to is called the zone of proliferation, or the zone of cell proliferation. And we find very small chondrocytes, or cartilage cells. Chondra is a root word that means cartilage. So very small chondrocytes that are in constant mitosis. So proliferation just means making more of. So these cells are in almost constant mitosis, and they're very small. Well, so we're making more cells. And as we make more cells, it's going to push some of those cells towards the diaphysis. So really what we're actively doing is pushing the epiphysis and the diaphysis away from each other. Now, as we're pushing those cells towards the diaphysis, the mitosis kind of starts to slow. And the cells themselves get bigger. So they're not going through mitosis, they're growing. And this is called the zone of hypertrophy, or the zone of cell hypertrophy. Now, in this place, like I said, we're not going through mitosis anymore. The cells are getting bigger. As the cells get bigger, the space that they occupy gets bigger. Meanwhile, we're still making more cartilage cells up in the zone of proliferation, pushing more cells into the zone of hypertrophy. The cells that were getting bigger in the zone of hypertrophy are pushed down to the next area. This is called the zone of calcification. And here, that cartilage matrix is going to start to become calcified. It's going to start to crystallize. And as that mitosis keeps going, and we keep pushing more cells into the hypertrophic zone, into the calcification zone is pushed down into the last area down here, the zone of bone deposition. So the osteocytes by this point have died. And we're going to start to have those lacuna surrounded by that calcified cartilage matrix. And we're going to start to break those walls of the lacuna down. And that leaves kind of open gaps. And those open gaps allow osteoblasts to come in. And when the osteoblasts move in, they're going to start to convert that cartilage into bone. Now that continues throughout childhood, throughout adolescence, into late teens, early 20s. And at certain times, there's going to be a very fast rate of this. And what's actually happening is, as you go through adolescence, the sex hormones that were previously not being released very much at all are going to start to be released at a really high rate. And as those sex hormones are released, they are going to cause this to happen really, really fast. And that's what we get with growth spurts. Growth spurts are the result of a few different hormones, but primarily they are the result of the sex hormones, both estrogen and testosterone. Now, as we enter into adolescence, we're having a real sharp rise in those sex hormones. So we have a real strong growth spurt. But then late in adolescence, those sex hormone levels start to taper off to a more normal level that'll kind of stay with us for most of the rest of our lives. And as those sex hormone levels start to taper off, that is what then triggers the closure of the epiphyseal plate. But it doesn't completely close. We are left with a scar in that area called the epiphyseal line. So the epiphyseal line is the scar where the epiphyseal plate used to be. So what we just saw was called interstitial growth. And interstitial growth, I mean, really, it's cartilage growth. That's what is causing that extra bone. But there is also something called appositional growth. And appositional growth is, instead of growth in length, it's going to be growth in width. The bone is going to get wider. And while growth in length tapers off and eventually stops, the growth in width can occur throughout your life. It occurs while the bones are getting longer, but it can continue throughout your life. And the way that this works is those osteogenic stem cells produce more osteoblasts on the outside edge of the bone. And they are going to lay down new circumferential lamellae. So the bone can get wider throughout your life. And we'll see what causes that in just a moment. There is something called Wolff's law, or Wolff's law of bone. And this says that the more stress that is placed on bone, the bigger and the denser that bone will grow. Another way to say that is bone growth and density is proportional to the amount of stress on that bone. Now, this goes both ways. The more stress is placed on that bone, the stronger the bone will get. The less stress that is placed on that bone, the weaker the bone will get. So, this can be seen in people who are weightlifters, people who are constantly lifting weight and putting a lot of stress on their bones. Their bones will be very dense, very strong. It doesn't even have to be a lot of stress. When we look at, if you are right-handed, if we compare the bones in your right arm and right hand to the bones in your left arm and left hand, the ones in your right arm will be a little bit denser, a little bit stronger, just because they experience more stress. This goes down to even at the different places, the different parts of a bone. One bone will have denser areas based on where the stress is. When you look at bones in lab and you see things like the points where muscle are attached, things like the trochanter, the greater and lesser trochanter in the femur, well, those are points where there's going to be more stress. And as I said, this goes the other way also. Patients who are bedridden, patients who are paralyzed, or if you've ever broken a bone, anytime you're not placing stress on a bone, it's going to get much weaker. And I always kind of get a chuckle out of when you watch one of those feel-good movies and someone wakes out of a coma and suddenly they're like, oh my gosh, I'm awake, and they get up and walk across the room to hug somebody. That would absolutely not happen. If they'd been in a coma all this time and they tried to stand up, they would shatter the bones in their legs because their legs would not be strong enough to support them. When you see people who are astronauts and they're up on the space station, they're oftentimes running on a treadmill and they've got this big, looks like a huge rubber band pulling them down against the treadmill. That's because if they didn't do something like that, there is no stress on their bones when they're in space. There's no gravity. They are simulating gravity by pulling themselves down against a treadmill and continually putting strain, putting stress on those bones. But even that's not enough, because if you ever see the video of astronauts who have been in the space station for any length of time, when they come home, they've always got someone supporting them because they need that extra assistance since they haven't been putting as much stress on their bones. All right, so that's where we will stop this lecture. We have one more lecture of this chapter left, one more lecture of this unit left, and that's going to be the physiology of bone. All right, so if you have any questions at all, let me know. I will talk to you next time. Take care.